Welcome to our Spectronaut 14 launch seminar. My name is Max Half. I'm a product manager here at Diagnosis, and I look forward to sharing with you what's new in the new version of Spectronaut and how it will reshape the DIA-based proteomics. And this seminar has 3 parts. So at first, I'll talk about Spectronaut 14 and what's new, and a little bit about iMobility support. And then Oliver Bernhardt, the lead developer of Spectronaut, will talk about SNE Combine, which is a new way to resource efficiently analyze large-scale experiments with Spectronaut. And finally, Lucas Reiter, our CTO, will dive deeper into DirectDIA 2.0, which is a major improvement over our previous workflow for library-free DIA proteomics. And you can ask questions through the chat functionality of this webinar program. So you can just ask at any time. We will collect all the questions and answer them at the end after all 3 of us have given our talks. I would like to take the opportunity to also mention some other great events that we have organized for you at this year's virtual ASMS. So we have some really high-profile scientists among our users, and we invited some of them to give presentations on their research. And on Thursday, we have Jesper Olsen and Florian Meyer. And on Tuesday next week, we are looking forward to talks by Birgit Schilling and Yancheng Liu. So yeah, make sure to sign up for that. The links are also on our website. And in addition to that, we will be at our virtual booth this Friday. So we will go over the new features in Spectronaut. There will be live demos, and you can also just chat with our developers there in a more informal setting. In addition to that, we have some pre-recorded talks that we gave at vendor events in the lead up to ASMS that are available online. That might interest you. There's also a workshop I would like to mention where Lind Weberke will be a panelist at a discussion about DIA that is hosted by Birgit Schilling and Florian Meyer on Wednesday. And we as a company are presenting a total of 8 scientific posters ourselves, and there are also some presentations from our collaborators. You can find all that information on our website at diagnosis.com/ASMS2020 Reboot. Now, I want to give you a little insight into our company. And at the core of our activities is a strong research and development department that develops new proteomics methods and ever better software solutions. We have built strong scientific collaborations with leading experts worldwide that help drive our progress here. And out of this strong R&D focus, we can offer high-quality software products on the one hand, like Spectronaut, and on the other hand, we can also provide state-of-the-art contact research services using our own mass spec facilities and our software as well. Diagnosis was founded in 2008 as a spin-off from the group of Rudi Aebersold at ETH Zurich, and since then it has grown into a company of about 50 people and offers a range of products and services for proteomics. Around 2012, with ETH and SciEx, we co-developed targeted analysis of data-independent acquisition for large-scale proteome studies. And we started to release our software solutions for proteomics data analysis in 2013. Of course, our flagship product is a feature-rich and powerful software to analyze DIA data called Spectronaut. And in 2017, we integrated the DirectDIA workflow into Spectronaut that enables DIA analysis without a DDA library. And that's something we will, of course, hear more about later. And with that, our latest milestone is the release of Spectronaut 14. Here's a little refresher on the differences between data-dependent acquisition, or DDA, and data-independent acquisition, DIA. In DDA, MS2 spectra are acquired for specific precursor ions, and this has the advantage that essentially all fragments you see at MS2 level come from one specific peptide precursor ion. However, this leads to a semi-stochastic sampling of the peptide population of a sample. In DIA data, on the other hand, a set of MS2 scans with a wide isolation window is acquired, and all ions in that wide range are concurrently fragmented. This makes it possible to get MS2 fragmentation for all precursors in a sample across the entire retention time range and allows quantification at MS2 level. But as you can imagine, the MS2 data looks a lot more complex, and you need specialized software like Spectronaut to deconvolute these MS2 spectra. Spectronaut, of course, is our solution for DIA proteomics, and you can use it for all steps of data analysis, from generating libraries from DDA or DIA data to DIA analysis and quantification, all the way to data interpretation. That includes discovery and localization of post-translational modifications and specialized workflows for labeling experiments. We are regularly improving and updating the software, so there's a new release with new features every year and regular updates for smaller improvements as well. And we have a highly qualified support team in place, and the developers themselves are in touch with customers as well to help them get everything to work perfectly. So we're really proud of our customer service, and this close connection with our customers is really important to us because that's how we know what people need for their projects. And that, of course, helps us to direct the development of new features for the software. Fortunately, Spectronaut has been well received by the scientific community as well as our industry partners. And we saw almost 200 papers in 2019 mentioning Spectronaut. So, as mentioned earlier, in this first part of our launch seminar, I will give you an overview of what exactly is new in Spectronaut 14. Now that we're done with some of the more general topics. So we have three major features that we would like to tell you about, as well as a range of additional improvements, which I will just touch on briefly at the end of this. So you will hear more about DirectDIA 2.0 and SNE combined from Lucas and Oliver later. And I will just start now by giving you a quick overview of our support for ion mobility technologies. So in most proteomics data, we have three dimensions: the retention time, mass to charge ratio, and ion intensity. The addition of ion mobility separation devices is an increasingly popular option to get another dimension of separation for mass spec experiments. And the collision cross-section, that's the physical property that causes differences in ion mobility, generally correlates with the mass to charge ratio. But as we can see here, it does add an opportunity to remove contaminants and can also help to separate peptide ions with otherwise similar properties. And this improves sensitivity and can be a powerful tool to improve proteome coverage. The major instrument vendors use different implementations for ion mobility separation. Waters has offered ion mobility with some of their mass spectrometers for years under the name HDMSE. And maybe the most important recent additions to the DIA proteomics toolbox are dia-PASEF by Bruker, which adds an ion mobility dimension to the data, and Thermo's FAIMS Pro, which essentially adds a filter that will only let part of the ions pass, depending on the applied compensation voltage. All three of these technologies are now supported by Spectronaut. So let's take a look at how we handle the four-dimensional data that we get from dia-PASEF. Unfortunately, we were able to reuse many concepts and algorithms developed for the regular three-dimensional DIA data here. One such concept is that of high-precision retention time, which allows Spectronaut to accurately predict where in the RT dimension you expect to see a certain peptide with what tolerance window. We now process ion mobility in a similar manner by creating an ion mobility calibration based on the values in the library versus what we observe empirically. And this allows us to account for any shifts in the ion mobility dimension, and we can still narrowly extract ion mobiligrams. For example, here you can see an XIC of a peptide in the ion mobility dimension during the analysis step. And with the high precision calibration workflow, the extraction windows can center around the calibrated ion mobility range with a narrow extraction width. And this yields great identification results, and you don't have to try different static extraction widths to optimize and get better identification. Spectronaut automatically gives you pretty much the optimal setting for this through this calibration process. And we also see good quantification results with dia-PASEF. So we see here data for a mixed proteome sample. So there were 2 sample groups with different relative proportions of material from these 3 species, human yeast and E. coli. And the expected fold changes between sample groups are shown by the dashed horizontal lines here. And we can see that the actual quantification results are nicely distributed around the expected ratios. And this is data from dia-PASEF runs analyzed by our DirectDIA 2.0 workflow, which means without acquiring any additional runs for library generation. We identified more than 7,400 protein groups per file on average here. This was a 100-minute gradient. This is from a different, slightly different data set, also HeLa cells. This time we're looking at FAIMS Pro data. This was a 120-minute gradient. And we see that in this case, we use Spectronaut to generate hybrid libraries from DDA and DIA data for the data analysis. And what we can see here is that adding FAIMS Pro lifts up the number of modified peptide IDs and identified protein groups significantly. And that allows us to identify close to an average of 10,000 protein groups per sample from these triplicate HeLa runs. We have more content for you on ion mobility here at ASMS. So there are 2 posters from Biognosys, one about dia-PASEF and one on FAIMS Pro. There's also a talk available online that my colleague Tejas Gandhi gave at the Bruker X-Seed Symposia very recently. And you can find all of this information as well on our website at biognosys.com/ASMS2020 Reboot. Now, let me briefly talk about some other changes to Spectronaut, our improved deep learning augmentation, and Quant 3.0. So what's Quant 3.0? In a recent paper, we teamed up with Olga Witek's lab to demonstrate an improved statistical test for differential abundance in proteomics DIA data. And with this new method, differential abundance is tested with intensity data from both MS1 and MS2 levels. This improved the quality of statistical testing results and showed to increase the coverage of known activated pathways in a benchmark dataset. This was originally an analysis script, but we have integrated this new test seamlessly into Spectronaut 14, and it is the new default option for differential abundance testing, so you can very easily take advantage of this improvement. And now I would like to touch on the different stages of analysis where we use deep learning. So first, during database search, we use some scores that are based on predictions from deep learning models. Then during IRT calibration, we use deep learning for IRT prediction. And thirdly, we have built a model to predict the fragmentation patterns of in silico generated peptide sequences so that we get better decoys for FDR control of our DIA analysis. These and other improvements have helped us to achieve a remarkable increase in proteome coverage. And as you can see here, we get almost 25% more identifications out of the DirectDIA workflow compared to the previous version 13 of Spectronaut. And we also see an 8% improvement in identifications from workflows using a DDA library. In addition to these large advances, we have added more functionality in Spectronaut 14 on top of that. We now support some of the most recently published acquisition modes, added visualizations that give you an easy way to make publication-grade figures, and added more options on reporting quantification results, including full details on quantifications for each channel in labeled experiments. You can also now take advantage of ETD or ETH-CD fragmentation, and it is now possible to export search archives from DirectDIA experiments. And this is the end of my talk. Remember, if you have any questions, you can ask them in the chat. We will answer questions at the end of the seminar. And with that, I'm very excited to hand over to Oliver Bernhardt, who will introduce you to the new SNE combined workflow in Spectronaut 14.
Thank you, Max. Yeah, as Max already alluded in his overview slide, so the topic of my talk today is I want to talk about something I was working on for the Spectronaut 14 release, and the title of my talk is Scalability Redefined: How to Analyze 10,000 plus DIA runs with Spectronaut. And immediately you can start off with the question, how do you actually analyze 10,000 plus samples in any DIA software, essentially? But I'm focusing on Spectronaut here. And I want to highlight that I am mostly talking about here from a technical aspect, so from the computational challenges that arise from that, mainly also the hardware challenges that you face when you're having a DIA experiment of that magnitude. So what are some options how you can analyze this data? So obviously the first thing that comes to mind is, just analyze it like any other DIA experiment, and just put everything, all the 10,000 runs, put it into Spectronaut, and just let it run through. And there are obviously some benefits for that. So you have experiment-wide FDR control, you have cross-run normalization that is enabled, you have global interference correction, and unified quantification selection, and also global protein inference that you can take advantage of. But there are also some downsides, obviously. So one of them would be that you need all of the raw files already acquired before you can start your analysis. And there is also some drawback when it comes to memory consumption when you're doing a single large experiment like this. How exactly would this look like? So I show here a slide from, it was still from Spectronaut 13, but still it's quite applicable. So in Spectronaut, the memory consumption of an analysis pretty much scales with the number of precursors and the number of runs. So I have two different examples here, one's with a library showing 200,000 precursors and one's with a library of 30,000 precursors. And you can see that for the 200,000 library, you have about 107 megabytes per run that is estimated for memory growth per run, or 23 megabytes for the 30K library. But in essence, you can break it down to about 700 byte per precursor per run for an estimate for the memory consumption or the memory growth in Spectronaut. But that also means that if you extrapolate this further and go to 10,000 samples and a 200,000 precursor library, that you end up with a memory estimate of 1.3 terabyte of RAM. So obviously that is quite some beefy hardware that you would need for that. So is there a different way how you can do this? Well, there is also the possibility that you say, okay, I run my DIA samples all individually. Benefit of that is obviously that you have very low system requirements for that, since the software only needs to handle one file at a time, essentially. And also you can essentially start analysis while you're still acquiring your data. So basically, as soon as the first run is out, you can start analyzing your data. Drawbacks, obviously, you're missing out on all of the fancy post-processing steps that Spectronaut does. So you don't have experiment-wide FDR control, you have no cross-run normalization, no global interference correction, or unified quantification selection, or protein inference done. So all of these steps would have to essentially be done by the user in post. So the question now is there a way where we can have the best of both? And that was something that we were also thinking internally. And what we came up with is what Max already said is our SNE combine pipeline. So before I start with that, I quickly want to give a short introduction to what the SNE file actually is. And so the SNE file, for those who have used Spectronaut in the past, they might be familiar with it, but it's essentially a saved experiment file for Spectronaut. And that contains like library information, scoring information, all the quantification information, and importantly also FDR and decoy information. So it's essentially a complete experiment in one file. And that allows us basically to retrieve all of the important information after, when we load an SNE file, like the raw file would have been loaded freshly from an experiment. So how can we leverage this SNE file now into a new pipeline? So essentially what we were working on is the SNE combine pipeline. So what is SNE combining? It's taking these saved experiment batch snapshots essentially and running them through a specialized new pipeline that we developed that builds a comprehensive FDR controlled and properly normalized and interference corrected result overview of the entire data set. So you get all the benefits from the single run analysis, that you have very little memory overhead, but you also get all the benefits from the global run analysis, like FDR control, interference correction, etc. So this is obviously quite an interesting and powerful new approach. And when we were developing this, we were also focusing a lot on scalability in general. So that was one of the main focuses for developing this new pipeline. So we were not as much concerned about runtime, but we wanted it to scale basically till the end. So using this new pipeline, there are two use cases that I could think of immediately how one could leverage this now. That would be for use case one, what I call sequential analysis, which is quite interesting, is where you have a long-running acquisition series, let's say over a month, if it's a really large project. And whenever there's a new file generated by the instrument, you can directly run it through Spectronaut, generate an SNE file from it. And then in the end, once your entire acquisition series is done, after the last run is acquired and then run through Spectronaut, you can immediately start gathering all the SNE files you have gathered so far and run them through the SNE combine process to get a combined report. So basically, a few days after your last run is acquired, you have the final results, even if you have a 10,000 run project. Use case two would be what I call parallel analysis. This is more the use case if you have the data already acquired, but you want to run it through as fast as possible. And there one could do a distributed main analysis. So you can split up your large experiments into several batches and distribute them over multiple different analysis computers. And each of them is generating basically the SNE files for small subsets of the batches. And then in the end, again, you can take all the SNE files that were generated and combine them into one large comprehensive result table. So I've talked about the focus on scalability. So I also want to show a benchmark for this. And for this benchmark, I took one of our larger internal studies that we had available. And there we used the library of roughly 200,000 precursors. And what I did here was I incrementally doubled, so I doubled the number of runs that I used for this experiment with each step, and monitored the runtime, and I monitored the memory consumption. Unfortunately, I did not quite make it to 10,000. At some point, I had to make my slides, but I managed to go all the way till 8,192. And at this point, so what is nice about this workflow is when you look at the time consumption, it scales pretty much linearly. So if you know how long it takes for 8 runs to combine your SNE files, you can extrapolate pretty well how long it will take for the entire experiment. But more interestingly is the memory consumption, which basically stayed pretty much flat over the entire size of the experiment. So you can see there's almost no difference between running 32 runs, combining them, compared to the whole 1,892 runs on a regular desktop PC. So in this particular example, the maximum memory usage I had for the largest experiment was a bit over 7 GB of RAM. So how do we do this now? So the SNE combine, I want to look a little bit under the hood. And for that, when we thought about this and how to develop this, as I said, we wanted to focus mainly on scalability. So on sacrificing speed mainly, but scalability was the focus here. And so we developed a pipeline that works in 4 different stages. And in each of the stages, the pipeline essentially loops once through all of the SNE files. So if you have 8,000 SNE files, individual ones, it would run 4 times 8,000 times through the SNE file, basically. And in the first stage of this pipeline, the Spectronaut gathers some overall library evidence information. It starts already with Z-score normalization, which is important for a later process. And we already can perform protein inference in this stage. In the second stage, we depend on the information from the previous tasks. This is indicated by the red arrows, which means that these tasks the previous tasks need to have seen all of the data before the next process can operate. So in the next stage, we have interference correction that we can start, and we also build an evidence score summary map, which is again important for the next steps. In stage 3, we can then use the previous information to tackle normalization, single hit annotation, and protein FDR. And then in the final stage, we can combine everything together in the data summary process that is then fed into the quant summary process and into the report export. So doing something like this, obviously, in order to have such scalability, there were some sacrifices that had been made. So there's some things that are not easily scalable to this degree, and this is what I want to focus on now on this slide. So some of the sacrifices we had to make for this pipeline is there is obviously, in this case, there is no data browsing via the user interface. But one would assume that once you reach a few thousand raw files, that you don't look at individual peaks anymore. Also, currently, this pipeline only works with the peptide-centric DIA analysis in the back. So you would have to have SNE files that were run with the library-based approach in Spectronaut. And also, the library that is used must remain constant over the entire length of the experiment. So you cannot easily switch your library midway through the experiment. Another thing that was a bit tricky to do, and that's why it was scrapped, so you will not find in the report output, you will not find CVs, especially condition-wise CVs, because they don't scale well. We also have no local normalization. For those who know Spectronaut, quantity normalization comes in local normalization or in global. And for the SNE combined pipeline, we had to fix it on the global normalization because, again, the local normalization doesn't scale well. And furthermore, there is currently no support for regulation analysis directly in this process. So the regulation analysis would have to be done then on the resulting spreadsheets. And pivot reports are also not possible in this mode, again, because the data structure you would need for a pivot report doesn't scale well. But for a long format report, you can provide pretty much any report schema that you can make in Spectronaut. Also important to note, we will continue to work on this pipeline, so be ready for more support in the future on this. We plan to extend this if it gains in popularity. But now I want to go back to my initial question again. So how do you analyze a DIA study containing 10,000 plus samples? Well, I don't know whether this is the best way, but this is how I would do it. And this goes back to what I showed before with the sequential analysis. So in this particular example, you would have your 10,000 samples that you want to measure. You run them obviously through your instrument, and then you take the recorded MS files and a spectral library that you have predetermined before, and you run it through your DIA main analysis. So one DIA file, so one DIA sample at a time. At this point, you could already have immediate QC feedback that you determine yourself in the beginning. So if you have a QC fail, essentially, you could immediately start reacquiring that particular sample because maybe the instrument needed cleaning or whatever. But if everything is good and passes your QC criteria, you can save the experiment as an SNE file and store it for later for the final SNE combined pipeline. Once the entire series of 10,000 samples was acquired, you can then immediately, after the last sample is done, shove everything into SNE combine and get your final report in the matter of hours to days, depending on how large your experiment is. And the cool thing about this is you can do this entire setup on a computer with essentially 16 gigabytes of RAM should be sufficient. So a medium-sized laptop could do this kind of large-scale study with SNE combined in Spectronaut 14. So how do you actually, do an SNE combined pipeline in Spectronaut? I don't have a time to go too much into detail anymore, but you can find the SNE combine on the pipeline perspective. There's a small link in the bottom hidden that is called Setup SNE Combine Process, and that allows you to set up one of these experiments. So now I already reached my summary slide. So quickly to reiterate, SNE Combine, what is it? It's our novel experiment-wide processing pipeline that allows you to do the DIA analysis as separate main analysis, which does the heavy lifting, and then combined experiment-wide processing that is implemented in a way that it gives you full experiment-wide FDR control, synchronized quantification, and interference correction and is geared towards extreme scalability with easily doing tens of thousands of single raw files on a consumer-grade desktop PC in the end. So with this, I want to thank you very much for your attention. I want to highlight two posters that we have. So one is my own, showing again this process. You can check that one out. And another one is from my colleague Jakob Vohwinkel, who used this new process for showing off his new project, basically in this poster. And with that, I want to hand over now to my colleague Lucas Reiter, who will talk about DirectDIA 2.0 and the improvements we made. Thank you very much.
Welcome everybody, and thank you very much, Oli, for the handover. I'm going to talk about DirectDIA 2.0 and how direct searching of DIA data caught up with using sample-specific libraries. Now, DIA data has been around in proteomics for roughly 20 years, but not always has it been that easy to analyze DIA data as nowadays. And here you can see that in the beginning of the DIA development, mostly what is called spectrum-centric analysis of DIA data was used. Here I have a couple of papers that I highlight in the early days of DIA that deal with DIA data acquisition, but also DIA data analysis. So they all use this spectrum-centric way of analyzing DIA data. And the reason why they did that was simply because during these times, this was the way how DDA data, data-dependent acquisition data, was analyzed. And hence, they just used the existing tools back then to analyze the DIA data. But then around 2012, our colleagues from ETH, from the group of Rudi Ebersold, Ludovic Gillet, they published this paper, the SWOF paper, that is about the targeted analysis of DIA data, or in other words, the peptide-centric analysis of DIA data. And around 2012, people started to analyze their DIA data using this peptide-centric way of analyzing the data. Now, what is this spectrum and peptide-centric analysis of DIA data? Let's first start with the spectrum-centric DIA data analysis. In the spectrum-centric DIA analysis, you start with your spectra and you query your search space using the spectra. Here depicted with these three, the search space is depicted with these three peptides. Now, in contrast to that, for the peptide-centric analysis, you start with your peptide in your, for instance, library, and you query your spectra using this peptide. Now, inherent to these completely different ways of analyzing your data, there are certain advantages. And one advantage for the spectrum-centric way of analyzing the data is certainly to deal with huge search space. For instance, a phosphoproteomic search space, or in general PTMs, or also other kinds of very large search spaces. But then it also has an advantage in detecting conflicting explanations for DIA data, and that's also inherent to the way the search space is queried with the spectra. Now, the strengths for the peptide-centric analysis, on the other hand, are certainly the speed, but also the quantification. And again, here, this is inherent to the way the data is being analyzed, that this approach is very well suited for quantification. At Biognosys, we have been using this workflow on this slide here for almost 10 years now as our default workflow for analyzing DIA data. And while out of experience, we know that this workflow provides high quality quantification, you can see that this workflow is relatively complicated. And it's particularly the upper branch of this workflow, the library generation branch, which make this workflow more complicated than it could be in principle. This is also why people have been working on the spectrum-centric analysis to make this more powerful and easier applicable. And here I want to highlight a paper from Alexei Nesvitsky's group, the DIA Empire paper, that is a great read about this approach. First, I want to show how DDA data is analyzed in the spectrum-centric way, and then I'm going to talk about DIA and what the actually minor differences are between the two. In DDA, MS1 spectra are being acquired, and the mass spectrometer detects MS1 features in the MS1 spectrum. These peptide features are being fragmented, and this fragmentation process can happen at any time point in the elution profile of the peptide. And then the resulting MS2 spectra can be queried against the search space and be identified. Now, in data independent acquisition, as you heard before, people use larger precursor selection windows for fragment the peptides. And often there are multiple peptides in these precursor selection windows that are fragmented at the same time. Now, the difference here is because there is a continuous record on MS2 level that you can actually look at the MS2 scans always at the apex of the peptides. And also you can do some pre-processing of the MS2 spectra, and then you can actually do a very standard database search similar to what is being done for DDA data. Now in Spectronaut 14, we have added something new to this pipeline, and this is deep learning. Deep learning has proved to be very powerful in making predictions across various domains, but among many domains also predicting peptide physicochemical properties. And I highlighted here one paper below that was one of the first papers that showed this. Now in Spectronaut 14, we integrated an in-house developed deep neural network, and I'm going to tell you now a few features about this deep neural network. It is a multitask deep neural network that can predict fragmentation and retention time at the same time and uses this information for scoring. Because we do multitask prediction, and also because we optimized this deep neural network to be very fast for inference on CPUs, we can do these predictions on the fly without slowing down the search. As I mentioned before, this is being done automatically, so the user doesn't have to deal with that, and it's done on the fly while a search is being performed. In the end, we also have native PTM support. So for instance, phosphorylation is supported natively from the deep neural network. And on the right, you can see the network in action. You can see a predicted fragment ion spectrum for a peptide in one of our software packages. Now we wanted to see how this search performs on real data, and we used a state-of-the-art platform, an Orbitrap Explorer is connected to a FAIMS Pro, and we acquired DDA and DIA data with 1 hour gradients, and we applied this new pulsar search algorithm with the deep neural network for scoring on this data. First, I'm going to show the results that we got from these 1 hour DDA runs. Here we could identify around 5,000 proteins in 1 hour. Next, we looked at the DIA runs. Here we could identify around 6,000 proteins in 1 hour using the pulsar that was integrated in the old Spectronaut release from last year. And then finally, we took the same DIA run and applied the new pulsar to this DIA run, and we could identify yet another 1,000 proteins more in this 1 hour DIA run. All of these analyses were performed as single runs to not have any bias in the analysis. So there is no alignment or similar happening in these analyses. Now this is very striking because we get a 44% improvement when going from DDA to DIA on this setup that I show on the left, but also we get a more than 20% improvement when we use the deep neural network scoring on the DIA data using the new pulsar search algorithm. Now we can also integrate this updated algorithm into the DIA, the DirectDIA workflow, and I'm going to show you here what DirectDIA exactly is. So you start off with your samples, you acquire those in single-shot DIA runs, and then you can perform the search that I have presented just before using the pulsar search algorithm and the deep neural network for scoring. A library is generated automatically from this, and this library is then reapplied to the DIA runs using a peptide-centric analysis, which gives you the DirectDIA quantitative results. Now this is essentially a combination of the best of both worlds. And this approach can handle huge search spaces, such as, for instance, phosphoproteomics. It can also detect conflicting explanations for data. And it's also a workflow that provides high quality quantification because of the second step and the targeted analysis in the second step. Next, we wanted to see how this workflow performs in a real-life experiment. And for this, we took a lung cancer study. These samples were derived from 12 individuals with lung cancer. These were 12 times healthy tissue as well as 12 times lung cancer tissue. There were also 2 cancer subtypes in this study. And we acquired the runs on a similar setup as described before, with two-hour gradients now. And on the next slide, I'm going to show you the ID breakdown. So this is how the analysis looks like. In light blue, there are the proteins that we can detect in almost any run of the study. In dark blue, there are proteins that are expressed specifically in certain samples of the study, and hence we only identify those in certain spectrum-centric analysis of the runs. And then in red, you can essentially see the protein identifications that are transferred to the other runs using the peptide-centric analysis. Here I'm showing you one candidate, and I'm not going too much into the biology of this candidate, even though it's an interesting protein that may distinguish the 2 cancer subtypes. But I want to highlight here that this protein has only been identified in certain runs using the spectrum-centric analysis, and these were the runs of the adenocarcinoma samples. And of course, such kind of proteins are the interesting proteins. So these are the proteins we are often interested, the ones that are only expressed in certain samples or certain subgroups of samples. And the DirectDIA workflow is very much suited to finding those and having them in your final analysis. Now, of course, we are also interested how this DirectDIA2 workflow performs in comparison to using project-specific libraries. And for this, we have been doing a quite broad analysis or broad evaluation on 13 diverse datasets. 8 of these datasets were published. For 11 of these datasets, there was a deep fractionation performed to generate project-specific libraries. And the data was acquired on various MS vendor platforms. Various samples were involved in the studies, and almost 30 runs were used to build the libraries, and around 30 DI runs comprised the study in the end. And we were looking at how the DirectDIA2 performs in comparison to using the project DDA libraries. If we look at the proteins, these 2 workflows perform almost the same. When we look at the precursors, DirectDIA actually performs a bit better than using the deep project-specific libraries. And when we look at the MS time, not surprisingly, there is the MS time is almost cut in half, since a significant amount of the time has been used to build these project-specific libraries. So with this, I'm already at the end of my talk, and I want to summarize that in the newest Spectronaut, we have integrated our newest pulsar search algorithm that comes with a powerful deep neural network, which significantly increases identifications. Maybe on a side note, but with this TNN augmented pulsar, we could identify 44% more proteins from single-shot DIA acquisitions as compared to DDA acquisitions. And this is both, for both, we used the faster-based search space, and this corresponded to more than 7,000 proteins in 1 hour for HeLa. So this is quite impressive. Then what I showed on the last slide is that DirectDIA now caught up with deep project-specific libraries in terms of proteome coverage and also quantification. And in the end, so this essentially renders extra runs to make libraries largely obsolete now for DIA, which is really great. And with this, I would like to go to the acknowledgements. I would like to thank the whole diagnosis team here in Schlieren. Then also special thanks to the R&D team and also operations who were directly involved in the data that I showed here. And thank you for your attention.
Yes, thank you, Lucas, very much for the presentation, also to Oliver. And we've received a lot of questions from the audience, so that's great that people are engaged. And yeah, I think I'll just start by asking these questions. We will just go through them and answer as many as we can in the time that we have left. So maybe the first question for Oliver. So someone asked, when will scanning swath acquisition be supported in Spectronaut?
So theoretically, it should work. The problem is I never got any test data. So also shout out to the community. If someone can provide me with test data for scanning swath, I would love to give it a shot. But it should, in theory, already work. But I had to make a lot of assumptions on how the data structure looks.
Mm-hmm. And here's another one about SNE Combine. So will it be possible to use the SNE Combine workflow in combination with a cluster using, for instance, Slurm in the backend to distribute the 100 files per node over the cluster and combine them later?
So right now, the SNE Combine, we haven't gotten around to adding a command line interface for it, but it is planned as one of the first updates for this. In general, yes, as long as the cluster runs Microsoft Windows, it should pretty much work, I guess. The whole thing is you can do your DIA analysis via command line, and soon you will also be able to do the SNE Combine via command line as well.
Okay. And then we have another question about SNE Combine here. How much of a difference do you see in FDR for sequential processing versus combining the files all together?
The question. I cannot cross tasks now. So I'm assuming it's about the two different use cases I showed, and the two different use cases use the same algorithms for also the same algorithm as the standard standalone analysis uses. So you would not expect any difference in the FDR between those, also compared to the standard analysis. If the question was about not doing anything and running the files one by one and just taking the spreadsheets, I haven't evaluated it in that regard, but it would probably be way above 1% if you don't control it specifically experiment-wide.
Mm-hmm. Yeah, we have another one for SNE Combine. So someone asked, the SNE Combine addresses cohorts with large numbers of samples. How does the SNE Combine deal with the inter-batch variations between data acquired on different batches that can spread through months? And is there any suggestion on making a good for batch correction?
So we do apply batch correction internally for our very large studies. Currently we have not integrated automatic batch correction in the SNE Combine process, but this is also something that we are planning for the future to integrate into this process. But right now, batch correction would be up to the user.
Mm-hmm. Yeah, we have a question here. When will Spectronaut 14 be available if we already have a Spectronaut 13 license? So all you have to do is to just restart the software, and an upgrade window should just show up, and you can use the internal updater and get Spectronaut 14 right now. Okay, then we have some questions here for Lucas. So is it possible to combine DDA libraries and DirectDIA data, and does this increase coverage, or does this run a risk of too many false positives?
Yeah, I think this is referring to the hybrid library approach, and we actually have a publication about that in Molecular Omics, where we also looked at whether the FDR can be controlled well with such an approach, and we came to the conclusion that yes, one can do that. So you can combine DDA data and DIA data to make hybrid libraries and apply them to DIA data while still controlling for the FDR. However, what we have also seen And what I have shown in my slides is that with the improved DirectDIA algorithm, there is not such a big difference anymore between using hybrid libraries and using DirectDIA 2.0. And because there are certain other advantages of using DirectDIA 2.0, we would actually recommend to using this. But it always depends on the experiment, and it's basically up to the user what the user prefers. And every data set is again a little bit different, so it's maybe best to just try out.
Yeah, thank you. Yeah, there's another question about Quant 3.0. So maybe also for Lucas. So peak area from MS1 tends to have higher magnitude than MS2, and will the combined Quant approach results be biased towards the MS1?
Yeah, that's a good question. Actually, the two levels of quantities, they are normalized as one of the first steps to not have any bias in terms of intensity. So this is part of the processing pipeline, that there is no bias towards MS1.
Mm-hmm. There's another one here. Can DirectDIA handle phosphorylation data?
Yes, it can. So as I have mentioned in my talk, we explicitly support phosphorylation for the predictions, but we also support many other PTMs. And there is no PTM where there would be let's say, a drawback of using the new Spectronaut as compared to the old Spectronaut. So we would expect improvements for all PTMs for Spectronaut 14, no matter what PTM it is.
I also want to quickly add that for the phosphor dataset, there is also this the publication from the Olsen group, where they also used DirectDIA alongside library-based DIA, and they came to a similar conclusion that DirectDIA works excellent also in phosphoproteomics.
Okay, here's a question for Oliver. So can Spectronaut create some result file format which can be opened in Proteome Discoverer?
Not to my knowledge. I don't know what the input format for Proteome Discoverer looks like.
Okay, and then we have some more questions here. So yeah, for Oliver maybe. So can you combine SNE files that were made with older versions of Spectronaut as well using SNE Combine?
Yes, that works also. There's just one thing. So for the Spectronaut 14, we also dramatically increased the loading speed for SNE files, which is one thing you would not take benefit of. So it would take a bit longer to do the SNE Combine, but it works as well. It's just a bit slower.
Mm-hmm. And how important is it to keep settings the same across all SNE files? And what happens if you use different versions of a library?
Yeah, so as I stressed in my talk, keeping the library constant is currently highly advised because of the assumptions that the process makes. We obviously want to look also into this because that would open up the door for DirectDIA as well. But right now, this is how it is. And regarding the settings, usually one, I would imagine, keeps the settings for his main analysis, like the XRC extraction and scoring, more or less the same. But it's still, there is no underlying problem if you would change the settings for your main analysis, because for the SNE Combine, the experiment-wide processes are anyway blind to those settings, and they wouldn't basically wouldn't care if you changed individual settings per run.
Okay, and here's more questions about DirectDIA. So for Lucas, so how does DirectDIA compare to using in silico libraries?
Yeah, that's a very good question. So both DirectDIA and in silico libraries try to address the same problem of having to acquire extra runs to build a library. DirectDIA implicitly kind of creates a library during the search process, whereas for the in silico library, this library is generated explicitly, and this brings certain differences to these workflows. One is that, for instance, for very large search spaces, such as phosphoproteomics, for instance, this is more difficult to handle with in silico libraries because they can get very large. Whereas for the DirectDIA, this is not a problem. Also, FDR control, if not implemented properly, can be an issue with these huge in silico libraries but there it really depends on how FDR control is implemented for the targeted analysis of DIA data. So I would say these are the two major differences of the two workflows.
Yeah, there's a question here as well about the kind of data sets that we used for the comparisons between DirectDIA libraries and DirectDIA, and if these are representative of real-world applications. And because I did that, maybe I should just quickly jump in here. So we looked at data sets with at least two different groups of biological conditions and multiple replicates per condition. So, and almost all of the DDI libraries that were used were fractionated. So, yeah, in that sense, it was pretty realistic. And we also looked at different sample types, cell culture, tissues, body fluids from different species. And we acquired, or we used data that was acquired with different instruments from Thermo and SciEx. So we covered a relatively broad range of scenarios that we think are quite representative of the real world. Okay, and I think with that we are pretty much running out of time. So I just want to quickly remind you that there are a couple more events that we have coming up. So on Thursday, 9 a.m. Central Time, 4 p.m. Central European Time, we have our first Spectronaut user seminar, the virtual exhibit hall on Friday, another Spectronaut user seminar on Tuesday. And you can find all this information online for everything on the new Spectronaut and information on where to get a quote or how to get a trial license. You can go to biognosys.com/Spectronaut. And with that, I would like to thank our speakers, Oliver and Lucas. And I hope we could give you some interesting information. And we look forward to hearing from many of you. Yeah, just reach out to us if you have any more questions. Yeah, and with that, thank you very much and goodbye. Enjoy ASMS.